second session of Chen-Ket Chai, and I would like to invite him and to each take their positions.
Ik heb de floor to you first, Martin. Dear candidate, dear Chen-Ket, I would like to ask you to present to us in no more than 10 minutes the objectives, methods and key results of your thesis.
Highly learned promoter, highly learned supervisor, Professor Hong Bias, of course, I'm happy to present my results. And so the title, as you can read, is Biomechanical Properties of Atherosclerotic Plaque Tissue. And I'm going to explain why it's important to know the mechanical properties of atherosclerotic plaque. I will start with explaining what atherosclerosis is. Atherosclerosis is a disorder of the arterial wall, which is causing a thickening of the arterial wall due to accumulation of fatty tissue and other components. And the result you can see here. This is a thickening of the arterial wall. It is a so-called atherosclerotic plaque. It can go so far that it intrudes inside the blood vessel, as you can see here, and it constrains the blood flow. So of course constrained blood flow is not very nice and not desired. But what is even worse is a so-called plaque rupture. So I'm going to explain what I mean when I talk about plaque rupture. But first we have to need a look at the atherosclerotic plaque itself. So this is a cross-section of an atherosclerotic plaque. You see here where the blood is, this is a lumen where the blood is going through, and it consists, the atherosclerotic plaque consists of different components. Here in yellow you see the lipid core, which consists of fatty tissue and other components. And it is separated by a fibrous cap, which is here visualized in green, separating the lipid core from the bloodstream. When I talk about plaque rupture, I mean the rupture of this fibrous cap, leading to direct contact between the blood and the lipid core. This leads to a reaction of the blood, which leads to blood clotting. It can go so far that the clot occludes the entire vessel, which you see here on the right hand side. It occludes the entire vessel, and ladies and gentlemen, in fact, this is the main cause of heart attacks and strokes. So of course this is not desired at all. So clinicians try to treat this by removing this plaque tissue surgically. And it has been shown that plaque tissue removal is only benefits 20% of the patients. So in 80% of the case, plaque tissue is stable. So not all plaques are unstable and prone to rupture. The problem is that the identification methods are not reliable. The methods to identify which plaque is unstable, which plaque is stable are not reliable because they're based on general parameters. The method we want to propose is computational modeling. Computer modeling allows us to look at the stresses, at the forces of the occurring stresses and forces inside the blood vessel. You can see here it's able to visualize stress peaks, and it's able to look at the forces directly, which is a direct cause of plaque rupture. So it might be able to use this to improve plaque rupture prediction. The problem is that if we want to use this, we do face several challenges. And I'm going to summarize them here. You see, if you compare this FEMM model to this cross-section of a plaque, you see that the plaque consists of different components and is highly heterogeneous. There are different kinds of plaque tissues, fatty tissues, stiff tissues like calcifications, and we need to identify these different components, the mechanical properties of these different components, to use them as input for a plaque model, to make the plaque model more reliable, to increase the accuracy, so it can be used for actual plaque rupture prediction. For this, we need to test those components locally. Another thing is, if you zoom into the tissue, you see that there's a high fiber alignment. And this fiber alignment also needs to be included in those computer models. And if you want to include the fiber alignment, you need to know the mechanical properties of those fibers and also the fiber orientation. So to sum it up, the goal of this research, of our research, was to improve plaque modeling by testing the mechanical properties of the plaque tissue components locally, to investigate the mechanical properties of the fibrous tissue, and also to visualize how are the orientations of the fibrous tissue. So I will start by talking about the mechanical properties, how we did the mechanical tests. We obtained plaque tissue from the hospital, which we then sliced to obtain those cross-sections which you see here. Those cross-sections were then placed underneath an indentation test device, and we were able to indent different locations of the plaque tissue. What happens is what you see here. We used a spherical indenter, we indented the tissue, and we measured the occurring forces Fz and the deformation dz of the plaque tissue during indentation. And the unique thing about the setup is we did it above a confocal microscope, which allowed us to visualize the fibrous tissue. And this is what you see here. This is a microscopy image during indentation. You see the fibrous tissue of the plaque. You see in red the indenter. So the indenter is moving towards you out of the picture. And what happens is when we indent the tissue is the deformation of the fibers. And please note that the deformation of the fibers is direction dependent. This is also very important to be included inside the plaque model. So using this experiment, we can measure the forces, the deformation of those fibers, and we did it at different locations. So again, you see here this is a cross-section of a plaque. You see in blue the remaining intima we tested, in red the shoulder region, so we had a particular focus on the cap of course, and in green the middle of the fibrous cap, and these are the values we obtained. So 6 to 130 kilopascal for the shoulders, 11 to 260 for the mid cap, 25 to almost 900 kilopascal for the surrounding intima tissue. This is the first time that someone was able to provide this kind of data, to provide data for the mechanical properties of a fibrous tissue in plaques. Now let's move back to the challenges, what I pointed out. Now we did address two issues. One issue, the tissue is heterogeneous. We did local testing using microindentation tests. We tested the fiber stiffness, the mechanical properties of those fibers. And which is now missing is, of course it's nice to know the mechanical properties of those fibers, but it doesn't really help if you don't know how those fibers are actually aligned. So the next step is to be able to visualize the 3D structure of those fibers. The method we used is diffusion tensor imaging. It's a method based on MRI. We used the state-of-the-art MRI scanner to perform this DTI measurements. This has been already done for different kinds of tissue. It has been also done for, we also have done it for healthy arteries, and the results you see here. This is already something which is already known. You see that all those fiber tissues in healthy arteries are neatly aligned inside the arteriole, oriented in the circumferential direction. This I presented you as a reference because what you see now, this here is a diseased tissue. And the diseased tissue, you do see a couple of fibers which are aligned in the circumferential directors, but the point is you see a lot of fibers which are here visualized in green, which are oriented in the longitudinal direction, completely perpendicular to the actual fiber alignment of healthy tissue. And this is actually, this is surprising data even for us. And it's also new data which now can be implemented in modeling. So to sum up the most important points of our research, we have shown that plaque tissue consists of fibrous tissue. We have provided the data for the mechanical properties of those fibrous tissue. We have shown that how the fibers are aligned. They are aligned in the longitudinal direction, completely perpendicular to what you would expect to compare to healthy tissue. And a very important point is this data is now able, we are now able to include this data, the mechanical properties, the alignment of those fibers. If we combine this with imaging data, we would be able to provide a completely new input for plaque modeling, for computational modeling of plaques, which now is, would be more reliable, which would be another step towards being a tool for clinicians in hospitals to predict plaque rupture and to predict heart attacks and strokes. Thank you.
Thank you for your summary and success with the defense.
Thank you.
Thank you. And now we should start a real session. In this committee, two members from outside the TU Eindhoven participate. These are Professor Daan from the University of Amsterdam, Amsterdam Medical Center, and Dr. Gijzen from the Erasmus University of Rotterdam. We would like them and the Institute for them to participate in this committee. And I will give now the floor to Professor Daan.
Thank you, Mr. Rector. Dear candidates, first of all, my Congratulations with this nice piece of work. It's hard work, and you have obtained very interesting results. And of course, it was a pleasure for me to read your complete thesis. But then, if I read through all the chapters, I see a kind of an optimistic view when you start with your thesis, with a nice overview of the literature. You do some very nice experiments. We will come into detail into that. And then you conclude that, at least that's my summary, that there is no real future for a biomechanical approach. And even you conclude that we should move to imaging. So, do you agree with my summary of your summary? And what went wrong? And then the real question is, if you would start again, knowing all the things you know now, what would be your new approach then?
Highly learned opponent, thank you for your question. So yes, I was starting very optimistic, of course, for this project also, because it's also a very interesting project. And the point was that, that the research we have done showed some new data, and unfortunately it showed also a new challenge, basically, or not a new challenge, but something which has been already known for quite some time, the spread in data. This is a very big problem which has been already there for 20 years, the spread in data. But we were able to basically show that the spread in data is due to patient specificity. So we need patient-specific data to be able to use plaque models. And
if I may interrupt you, why do you say patient specificity? Because if you take two pieces of the same part of the fibrous cap, you've done that for instance in chapter 4, you will find even the same spread of the data, isn't it? If you test different parts of the fibrous cap. So there is also heterogeneity within more or less the same area of fibrous cap. So that is within the patient.
Yeah, this could be because of it could be because of the heterogeneity. That's one of the big issues. So, for instance, what Epenstein et al. Have shown with they tested the tissue even more locally than we did. They used a nano indentation, and they have shown that the classification methods, which have been now used, for instance, saying like this is calcified tissue and this is fibrous tissue, that maybe we need a more refined element, because even for calcification, the different degrees of calcification have shown that even for like one classification you have a wide spread of data. So it depends on the degree how the amount of calcification occurred. So this might be also one of the reasons why we found such a big spread.
So if you would restart again, let's say go back in time 4 years and do the same project, what would be your approach? What would you do different?
This is a good point. It's a good question. So if I would have restarted the project with the knowledge we know now, I would say we need to be able to make the mechanical test more standardized with better classification methods. What I just said, be able to really identify what kind of tissue we test, because like it was kind of crude how we did it with a micro-indentation test maybe.
Yeah, but the reality is that the tissue that you investigate is by definition is very heterogeneous. So did you select the right patient groups? Because you selected patients with symptomatic carotid disease, carotid artery disease. So they have huge plaques, they have intraplaque bleedings, it's a mess.
Yeah.
And then you start measuring in this mess and you find a mess.
Yes, this is true. The problem is to obtain the tissue. So, of course, it's easy to obtain this because patients who are asymptomatic obviously don't, or they might not go to the doctor, and people might not look at the atherosclerotic plaques because they don't show any symptoms. So it's harder to get these kind of data because even though those patients have plaques, the amount of stenosis might not be big enough to actually show this. But it might be an interesting approach to also test these kind of patient groups. Although I do think it's very difficult to obtain those big tissues.
Okay. Can I ask another question? So let's zoom in then on figure 4.1, I think, where you give us the details of where you selected your tissue from. Yeah, that's on page 54.
Mm-hmm.
So and you also showed that picture in your 10-minute talk. So this is quite essential, because this is the figure where you illustrate where you selected the tissue from. And that is, of course, goes along with your theory or your knowledge of where the plaque actually ruptures. So can you explain that in your own words? Where you believe that the actual rupture takes place in a carotid artery, not in a coronary, but in a carotid artery.
Yeah, we had, of course, a big focus on the fibrous cap, and we wanted to test the fibrous cap. And so we focused on shoulder regions and mid-cap regions, because we know that the shoulder regions might be a location where rupture occurs, but also the mid-cap regions, and that there's also some uncertainty. So we wanted to see if there are mechanical properties which significantly differ between between the mid-cap and the shoulder region.
So you know that I'm a pathologist, and I look a lot at these plaques. So what is your reaction if I say that most of the ruptures are not really enormous ruptures, but are small dissections where the shoulder of the cap loosens from the rest of the tissue. So exactly at the spot where the red and the blue comes together.
So that was actually the main reason why we wanted to test those shoulder locations, because we also thought that those locations might be very prone for rupture. And
but that's too easy, because my theory would imply that it's not the tissue itself, it's the link between the tissues.
Yeah, yeah, we tested, that's also the reason, we tested in that area also several locations, in the shoulder regions, also in the intima regions,
right.
And we could not find any significant difference for the mechanical properties. There's a point, of course, I'm talking about mechanical properties and not about failure properties, right. So we did not go far enough to go into failure. This is actually, that's another chapter where we tested it.
Yeah, and there you didn't isolate those pieces separately.
Yes, we didn't do it. That's true, because we tested it in the radial direction, and we were not able to test it in the shoulder regions because just the amount of tissue, the size of the tissue, didn't allow this kind of measurements.
Okay, thank you for your nice discussion and your answers.
Thank you.
To the record.
Thank you. And that session will be continued by Dr. Stevens.
Thank you, Mr. Rector. Dear candidate, Dr. Cohen-Wennes, let me start with congratulating you and your mentors on the completion of your PhD project. It's a great piece of work, I think, which has increased our understanding of the biomechanical properties of atherosclerotic plaques. I think it's an important step in understanding why these plaques rupture and why they might not. So you address a lot of issues, but there are still, of course, some questions remaining that I was wondering whether you have answered. So my first question is maybe a little bit naive question concerning your first chapter, page 8, where you show a computational analysis of the stress in a plaque. I think in this case it's in a clinical literature where you look at a coronary plaque, but suppose it would be a carotid artery plaque. And as I understand it, the way you do these measurements or these calculations is that you increase the interluminal pressure, blood pressure, and then see what the stresses are using appropriate tissue parameters. Do you think, is that the only reason a plaque would rupture as a consequence of these stresses? So what happens, for example, if a carotid plaque, I hope I don't have one, but it could be, if I do this or that, or when I turn my head, is there torsion and strain induced by these movements, and how big is that in relation to the stresses that are, or strains that are induced by the
most learned opponent. Thank you for your question. So this is actually also a very interesting question, how that how the movement of your head influences the mechanical properties of your carotid artery. To be honest, I haven't looked into the head movement a lot, but I do think that I'm not sure, but I don't think that the stretches of the arteries, well, if you don't do it very extreme, that it's so much that it will have a big influence on the strain of your artery.
Yeah. Is there anything known about when most plaque ruptures occur? Is that during the day when people are very active, or is that during the night when people are not so active?
Yes, that's. I think that depends on the type of the plaque. So it has been shown, for instance, that during exercise, a lot of plaque ruptures occurs at a certain location of the plaque. So I think it was the shoulder region. And during the rest, you have a lot of plaque ruptures which occur at the middle of the fibrous cap. But it has not been understood completely yet because the failure mechanism of plaque rupture has not been investigated enough yet to be able to explain these kind of results.
Do you think that's related to blood pressure, or is it related to?
It appears that this might be related to blood pressure, because if plaque rupture occurs if you do exercise at a certain location and then if you are at rest, it appears at another location, it shows that the stress distribution might be very different for those different kind of plaques. So the problem is that it is very challenging to look at those stress distribution, because the tissue, again, as we discussed with Professor Dahm, is that it is very heterogeneous tissue, and there are a lot of different factors which play a role. For instance, if you have a calcification and a soft tissue next to it, that there could be some kind of a debounding. But on the other hand, it could be also that just the collagen fibers, which the fibrous tissue which we investigate, that those rupture. So the point is that we don't understand the failure mechanism yet, so it's hard to draw these kind of conclusions. The only thing we can tell that during rest and exercise, different kind of location rupture.
I have another question for you. I want to talk a little bit more about the idea of bulk properties versus a weak link in the plaque. Because you mostly measure bulk properties of the tissue, and the idea is to put that in a model. How would you be able to model, or actually to measure, whether there would be weak links in the plaque that actually cause most prone to rupture?
This is also a very good question. So I think it's hard to measure this directly in a patient. I think there might be other approaches, like looking at the surrogate markers, like biomarkers, because atherosclerosis is connected to a lot of to inflammation, and a lot of proteins come free. And it has been shown that, for instance, MMP9 is connected to collagen degeneration. And this might be something you could look into. In fact, in our work, in our TAROS project, there were groups which were looking in the molecular imaging.
So that's back to imaging?
Yes, it would be back to imaging. But it's again, this is also very challenging. I'm not sure if it's very promising. Actually, what I wrote at the end, the conclusion, I think the most promising part might be looking at strain imaging, at least to be able to identify stable plaques and to reduce the amount of unnecessary surgery.
Well, then I have a question about the future imaging, of course. Chapter 5. You focus a lot on the fiber orientation that you measure with DTI. But of course, DTI is able to give you more metrics than only principal eigenvector. Can you explain a bit what other metrics would be perhaps of interest that you can derive from the tensor?
You mean for the fiber orientation, or?
Well, not specifically for fiber orientation, or at least maybe that gives you parametrics that can give you more information about the microstructure apart from the first eigenvector?
Well, yeah, the problem was we were not, at least I didn't really look into that because we were more interested in the fiber direction because it would have given us, it gave us additional value for the anisotropic behavior which we wanted to be included in models.
Let me help you with that. Let's, what do you measure with diffusion?
Well, the diffusion of a hydrogen of protons inside the tissue.
So, did you look at the diffusion itself, the magnitude within the different plaque components?
Look at the diffusion itself. You mean the amplitude of diffusion?
Diffusion constant?
We actually I always kept it, tried to keep it constant to make it comparable between the results. So I didn't really.
Diffusion is something that's a tissue property.
Yeah.
The viscosity of the water within the tissues. There's some people that are comparing diffusion weighted energy to add more plaque component information to the models. Similar is true for the fractional anisotropy. Do you know that?
Fractional anisotropy? Yeah, I didn't really have to look at those data. I did see that that there were different locations, different kinds of anisotropy depending on the location, and some locations had a very low anisotropy. So I assume that those locations are isotropic, but they could also be calcifications, that there is no fibrous tissue. But since I didn't really look closer at the morphology of the tissue and only on the fiber orientation, I'm not sure about this.
Now, the reason that I ask is because, I mean, it's really a mess in the blood, right? The fiber orientation. So how sure are you that this really represents an actual fiber orientation, or is merely an expression of the fact that there is a kind of chaotic orientation?
I am relatively confident about it because we also double checked that with the histology, which you also see in the image. So you can, I mean, of course, because we sliced the tissue, we could not see how the tissue complete orientations because we also sliced basically through some of the tissue, but we did see that the longitudinal alignment is present also in the histology, which is supporting our results of the DTI.
Do you think we can? Question. Do you think this is a feasible method eventually in vivo?
With the current methods, I don't. With the current, because currently I don't think so, because we needed with the state-of-the-art MRI machine, we needed to keep. It was an ex vivo test. The tissue was taken out of the patient, and we had to embed it. So it was completely embedded in agarose and not moving at all. And we had the tests were done by Dr. Chen-Ket Chai, and they were done over the weekend, over the night, without any movement of the tissue. So I don't think a patient is so patient that he cannot move it for one night. But maybe in the future, if new methods detail.
An advice would be to also look at these other metrics, maybe, because these might be easier to obtain in a living person than a full tensor at such high resolution. So it might be a way.
Yes, that's true.
Thank you for clear answer.
Thank you. Then the position will continue by Professor Vossen.
Thank you, Rector. Dear candidate, let me first congratulate you with the nice piece of work you performed in the last 4 years and the nice way you presented that in the thesis. A little bit inspired by the discussions up till now, I had 2 questions in mind, but I will add a third one. I start with that. It's a more philosophical question maybe, and that refers to what you write on page 9, or actually it starts at 8. They stated that although it's well established that the atherosclerotic plaque tissue behaves mechanically anisotropic, most studies, and model studies I guess, work with computer models assuming isotropic behavior. And then the sentence there, this simplification has negative influence on the accuracy of plaque modeling. Is that a general statement you would? So what makes a model accurate?
Hi, Lieutenant Opponent, thank you for your question. I'm happy to answer that question. It is It is, of course, a bit blunt to say like it's a simplification, because we actually don't know how big the influence would be to the assumption of isotropic behavior. So this is something which needs to be investigated. We would need to have a model including the anisotropic behavior and see how big the influence is. However, it has been shown by a study group by Tang et al., if I'm not mistaken. They have tried to include anisotropic behavior, but without really knowing how it looks like. They kind of guessed that it's more or less the same with healthy, and they showed that there is 5 times different in stresses. So it might be that the stresses for also for the disease tissue, including our results, that it might be also significantly different.
But I do agree with you that with making models more complex, you probably can make them more accurate.
Yes.
But if you make models more complex, you also have to deal with more parameters. And in these models, there are also parameters involved. And it depends on the accuracy which you can measure or determine these parameters. If this more complexity will indeed lead to more accuracy. So it's not always the case that complicated models are more accurate. That's what's
Sure.
and that is a step to my second question. Actually, it's about chapter 2 and 3. There we learned that in chapter 2, it's a very nice review of what has been done in the field. And there we learned that each investigator you mentioned studied the properties of plaque in a different way with different techniques, and also made more or less his own classification of the kind of tissue he was looking at. And I will, page 15 for instance, it's called cellular, hypercellular, calcified. 3 classifications. Another author, page 16, non-fibrous, fibrous, calcified. Page 17, atherosclerotic, fibrous, calcified. Page 18, calcified, echolucent, and mixed. Page 20, hematoma, fibrous, partly calcified, calcified. And you can go on like that. So that, and indeed you, at page 24, you say, it's impossible to draw definitive conclusions. An accurate classification could be beneficial. So that was a nice end of chapter 2. And that, okay, now in chapter 3 you will come up with an accurate or classification that will be beneficial. So I read chapter 3, and then you start with a new technique, so another technique, and a new classification. Aligned fibers, structured collagen, non-aligned fibers, unstructured collagen. So you added an extra classification. So can you indicate why this new method and this new classification is not just another approach, like the others, and will be successful and beneficial?
Yes, thank you for the question. The point of that chapter was actually the added value basically of that chapter was because we tested the mechanical properties locally, and we wanted to test different components of the tissue. This is something which has not been done before. So that's true. Then we added another classification method, but we also wanted to look how different are those different components. Different components. So that was the point, and that was the point. This has not been done before, and we thought that this might add some value to the literature.
So that means that another PhD student in the future that will make a review of the literature will put you against all those others and say, but this is a different way. That's because, and then your answer is that you looked at the different properties. But to be able to do that, you need histology to find out where. And if you go to the final idea about using biomechanical models, you want, of course, to classify whatever classification you take in patients, and then maybe use so there's no histology available. What would be a way to still handle this difficulty, or is that the end of the story?
So I think there are some maybe promising approaches in imaging also to look at the different components of a plaque tissue. For instance, iris or near-infrared spectroscopy. They are able to, near-infrared spectroscopy, if I'm not mistaken, is able to visualize the lipid pool very more or less nicely, accurately. And this might be also an approach to identify the different components. Also, also OCT might be also something which is able to visualize the local, due to the strain, the different kinds of strain patterns you obtain, you might be able to identify different components of a plaque tissue.
And it's all intravascular kind of.
Yes, that would be
There would
intravascular.
no non-invasive method to be able to do that.
I think non-invasive methods might be a bit tricky. I think the only one I remember now, which is also just started, I think, is the photoacoustic approach to visualize the behavior. So it uses the advantages of a light and combined with the advantages of ultrasound. And this might be also a method to ex vivo, so to non-invasively measure the strain and morphology of plaque tissue.
And we discussed it already with the previous opponent that MRI would be nice, but a little
Yes, that would be
too,
also,
of course, but that can change, of course. And that brings me to my last question in chapter 5, where you use these DTI, and I don't want to ask about DTI itself, but for me it was not clear, and probably because you talk that DTI, you made DTI images of the carotid plaque. But for me it was not clear what you mean with the carotid plaque. Because if I look at figure 5.1, I see a complete carotid, okay, this is a healthy one. And then in page 5, of figure 5.2, 5.3, I don't know exactly what I see. So can you explain me, because I have to interpret a little bit the results, what did you image actually? What part of the artery, and is it the plaque or is it whatever kind, did you image?
Yes. So we imaged the specimen which we obtained from the surgery, from the atherectomy surgery. So we obtained the tissue, only the inner part, the diseased inner part of the carotid artery, and this part we have been embedded in agarose and imaged using DTI. So it's not a complete artery like you see in 5.1, it's actually a complete artery. You can see it, some of the remaining tissue was actually stuck, that's the reason why you see some of them. Some of the fibers are not nicely. But for the other one, it's only the inner diseased tissue. So the outer part might look a little bit different.
But if I look at figure 5.2, where's the plaque? Or is it complete plaque?
This is the complete plaque.
Okay, just the plaque.
Yes, just.
Okay, so that's only part of what we see in 5.1.
Yes.
And then you see different called the fiber orientation. Could you speculate how this could relate to rupture? Because of course we know that collagen will orient due to forces or strain or whatever, but because you find quite some surprising results, but does it mean that you know a little bit more about rupture?
It's a bit hard to say something about rupture based on those results. It is interesting that those fibers are reoriented in a longitudinal direction, and I actually speculated that this might have something to do with the change in strain pattern, that it might be now more into longitudinal direction, and that would cause the realignment. For rupture, it's interesting because what we found, for instance, in using the confocal imaging, that most of the strain for this collagen stiffness, for the collagen fibers, occur perpendicular to the fiber alignment. And so we thought that maybe rupture would occur mostly perpendicular fiber alignment. And so maybe if the fibers are indeed oriented for plaque tissue like this, that if you have a lot radial strain, that then the rupture, the cross links of the fibers would go off and rupture might occur. But that's also only speculation.
So more random orientation would be beneficial for non-rupture, let's say in that way. Is that true? Or?
I'm not sure if that might be true. Well, if a rupture would really appear the way what I just said, then it might be more beneficial. But then usually fibers do align in the direction of where the strain occurs. So I don't think that.
Yeah, but that's for the case that they work as they should work, and they align and it's better to rear forces. But for rupture, maybe this alignment would not be the best way.
Yes, that could be true. But then again, then we come back to the issue that we don't really know why plaque rupture occurs. And there might be a lot of other factors which were mentioned, which play a role, and which might also have an effect on the structure.
Okay, so to finish, to come back to my first question, this doesn't mean that we need to make the model even more complex in all cases, but maybe in some cases we do. Thank you.
Yes.
Thank you. Then now, Gijs will have a discussion with you.
Dear candidate, also I would like to congratulate you with this work. You brought a challenging project to a good end. I want to include your co-promoter and promoter in these compliments. I want to follow up on the discussion on the orientation of the collagen fibers in the plaques. Indeed, what you demonstrated is that at least in 4 out of 9 plaques, the collagen at the luminal side is oriented in an axial direction, which is kind of surprising, and as you indicated, it is counterintuitive more or less. The discussion, you speculate a bit about why this happens, and you speculate along the lines that that these collagens orient in the direction of the main strain, which is the hypothesis for the healthy arteries. Which implies that in these plaques, during their development, the main strain direction should change from a circumferential to an axial direction. To support this speculations, you refer to a paper of McCormick. I looked at this paper, and it's basically a paper that describes a new ultrasound strain imaging technique, basically focusing on this technique and demonstrating that it works in 3 or 4 patients. So I guess I would not call that proof that axial strain actually occurs in carotid plaques. It's anecdotal evidence, not proof. As a mechanical engineer, I just wonder what is happening with those plaques to make the strain go from a circumferential direction due to the loading of the infraluminal pressure to a strain that is occurring in the more longitudinal direction. What is happening mechanically? Could you explain it to me?
Most learned opponent, thank you for your question. So I think that the strain pattern changes, or I think maybe it changes, and one explanation for that might be the change of the structure of arteries during the development of atherosclerosis. So as you know, a thickening of the artery occurs. So at the beginning, if you have a healthy artery, the artery is the wall of the artery is very thin and elastic, and due to atherosclerosis, the artery stiffens and thickens a lot. And also the thickness of the arterial wall also in the radial direction increases. So this might give more resistance to the blood pressure. And maybe due to this more added resistance, the strain pattern might change in the longitudinal direction. At least that was kind of what I speculated.
I agree with you that due to the increase of wall thickness and assuming that there are still load-bearing structures inside the wall, that your compliance of the whole structure will decrease, which will probably induce lower strain values, but why should it go from a circumferential orientation towards an axial orientation? That's the intriguing part, I would say. Have you any clues on why that might happen?
To be honest, I'm not sure. So I thought it's added resistance in the circumferential, I mean in the radial direction, and also if you have stenosis, which is very advanced, which we actually tested, advanced plaques, that there's also a lot of resistance possibly in the circumferential direction. And then I thought I speculated that the only direction which might be left is in the longitudinal direction.
Okay. So it's still unclear about
it is unclear, and I think the increased stiffness might, for instance, explain why we found locations which were where the orientation was a little bit more isotropic, more random. This might have been due to this increased stiffening and this increased heterogeneity of the tissue.
To follow up a bit on your hypothesis that the increased thickness might lead to reduced strain and therefore maybe more axial strain distribution might be present, so that effect will be stronger for thicker plaques.
Yes, I would think it might
Did
be.
you, and four out of 9 did show that feature very clearly, the 5 others did not. Were the 4 ones with the more axial orientation, were they thicker? Did you look at that?
That's a good point. I did not look at that, but it would be interesting to look at that.
Okay. One final question, if I may. Of course, I'm interested not only in plaque mechanics, but also the effect of blood flow on plaque composition. Do you exclude an effect of shear-induced impact on the endothelium on collagen distribution? Do you think that that might play a role in the distribution that you found as well?
Yes, I think what, for instance, also McCormick have shown that there is, due to atherosclerosis, a lot of shearing occurs inside the arterial wall, and this shearing might also add to the alignment pattern of the fibrous tissue.
But I mean more of the signaling effect of the blood flow. Do you think that that might influence, because it's specifically at the luminal side, which is of course close to the interface with the blood, might there be some effect of the blood flow?
Yeah, I think it might have an effect on the blood flow. Might have an effect. So you say you basically say that the flow changes, and the anterior cells basically feel this change in flow and basically signal the smooth muscle cells to produce collagen fibers in a different direction.